Hi, thanks for joining me today. We are going to talk about the human immune system. We're going to cover this basically by looking at the two branches of the immune system that are typically referred to, the innate, non-specific immune system that helps us to deal with pretty much any foreign object or pathogen that enters the body, and the adaptive immune system, which allows us to remember and have more specific responses to individual pathogens. Now, I think what's useful first is for us to watch this movie. And what you can see is a human white blood cell. This is a phagocyte. Is chasing around the little black dot, which is a bacterium. And oh, there he goes. He has been successful in his quest. Okay, so that took a while, but one thing that video nicely illustrates is that, that white blood cell doesn't necessarily have to know specifically the identity of that bacteria or have seen it before in order to chase it around. So what we're going to talk about next is what the innate immune response actually is. So as I mentioned already, it's non-specific. So it's sort of a response to anything that comes into your body that can be detected as foreign at all. And it consists of two different, what we call first line and second line defenses. And you sort of want to think about this as defenses that are working from the outside in. So if you think about what, this outside of your body is, the first thing that a pathogen would encounter usually is the skin. Now, in addition to that, on our skin and also in other parts of our bodies, we have a lot of bacteria already. And that bacteria, the normal microbiota, is there to protect us because it takes up the real estate that pathogens might use. And so these normal bacteria are using up food that a pathogen would want. They're taking up space, but they don't hurt us. They're harmless. In some cases, they help us. As we have already discussed, they can help us, for instance, by making vitamins. There's also mucosal secretions. So in addition to parts of our body that are covered with skin, we of course do have some areas of the body that are a little more vulnerable, and that's because we need to receive things. Either we need to receive things because we need to take in information, so through our eyes, or because we need to take in food, so through the mouth. So there are parts of the body that are a little bit more vulnerable, not covered with skin, and those are mucous membranes, of course. But in order to protect those mucous membranes, there are chemicals that we secrete that are antimicrobial in the fluids that keep those tissues moist. Now the second line is, slightly inside your body. So if something sort of sneaks past your skin, gets into your mucous membrane, is not deterred by the chemicals there, then you still have phagocytes. And that is, the word is derived from the term eat. And basically, as you saw in that movie, they will chase things around and take them up and digest them. In addition, in the bloodstream and tissues, there are antimicrobial proteins. And besides all of that, and this is something you're probably pretty familiar with actually if you've been alive long enough to have ever had an infection of any kind, if you, say, have a local infection, you'll get inflammation. So when you get that paper cut and it turns red, that's actually part of your body's normal response to try to clear whatever pathogens might be coming into your body at the time. And then we also have a more systemic and separate response, which is to raise the body's temperature to try to kill anything that might be in it that it shouldn't be there. And of course, this is a little bit risky. And it's kind of like the compromise you make when you take chemotherapeutics, which is that you're trying to kill the pathogen before you yourself are affected. Now, besides sort of this more general overview, there's one part of this that I'm going to focus on now, and that is a subset of antimicrobial proteins called the complement system. And these are very important. If you've read your textbook, then you know there are multiple complement proteins. So ask yourself at this point if you have already figured out how many there are. There are 9, and they have these really easy to remember names. They're called C1 through C9, C1, C2, C3, C4, C5, and so on. Now, the important thing to remember about these is that they are floating around in your bloodstream in an inactive state. And so they're activated by a number of different events or circumstances, which we'll discuss in a second. But they're normally just present, and you can sort of think of them as being this like large piece that, and this is in fact pretty accurate, these larger proteins that in order to activate them, something has to happen that causes them to be cut. Okay, and so these smaller pieces are then liberated and they're actually active. Okay. Now, there are 9 of these complement proteins, but hopefully from your reading, if you've read ahead, then you may already have a sense of the answer to this question. But of these 9, are they all equally important? Hopefully the answer that you've come up with is no. Or perhaps you just guessed that based on my tone of voice. But what if in fact one of them or several of them are more important? Which one would that be? So we are going to cover that as well. So C3 is known as the linchpin. It is the most important complement protein. If you have a defect in C3, it causes a pretty severe immune deficiency. And that is because all of the different ways that complement can be activated and all of the outcomes of complement rely on C3. Some of the other parts of the complement protein complex and pathway are sort of have very specific roles, and so you would, if you had a defect there, you would lose some specific component of the innate immune response. But if you lose C3, you kind of affect everything. Okay, so it's a much more serious problem. So let's talk now about the effects of the complement. We'll also, in a second, we'll talk about how to activate the complement system, because as I already mentioned, it's inactive. But what happens when you turn this, when you sort of initiate this response? So the first thing that happens is you get inflammation. Okay, so again, if you get that paper cut, or you have a small wound on your hand, or you scrape your knee, that the redness that you see, that inflammation, is in fact your body's response to recognizing that something foreign is in your body. So inflammation is one response. Something that I will abbreviate as the MAC attack. Okay, sometimes people find this easy to remember. The MAC attack basically is that some of the complement proteins get together and make holes in pathogenic or foreign cells, things in the body that shouldn't be there. They make holes in those cells, and that is a really bad thing. You can imagine, we've talked a lot about how all cells have a plasma membrane and some of them have a cell wall, and that integrity of that outer layer is really important for survival. Okay, so if you start poking holes in the cell membrane, then the cell is going to die. It just, you poke enough holes in it, it can't really survive that. So there's something called the MAC attack. And a third thing is called opsonization. So again, if you've read ahead, I'm just going to give you a second to sort of try and reach back into your mind and remember what that is. Okay. For those of you who find this term unfamiliar, opsonization is when a foreign body or microbe or pathogen is covered in antibodies. And antibodies, as you probably already know, are part of the adaptive immune response, a way to recognize and clear infections that are specific to the particular causative agent. And so already we can see that now there's like this link. Even though we're talking about innate immunity, the non-specific response, it does have links to the acquired immune system. Okay, so this is one of those links. Opsonization, though, once you have targeted something and covered it in antibodies, then that is just a general flag, okay, for phagocytosis. So if you opsonize something, then in that video that we saw, most likely that bacteria that was being chased around was covered in antibodies, and so it was now a target. It was like you covered it in big red lights, and all of the white blood cells that are capable of chasing down and digesting organisms are going to chase down and digest anything they see that has been opsonized. Okay, so those are the three different outcomes of setting this complement protein machinery in motion. We're now going to talk about three ways that we activate it. So how do we get that to happen? And there are three different pathways. We're going to talk about them all, but they have these names. So one's called the classical pathway, one's called the alternate pathway, and there's also the lectin pathway. Okay. So if we just sort of think about what is happening in each of these cases. So let's first deal with the effects of the complement. Inflammation is essentially a lot of stuff comes to the area. Okay, so this relies on white blood cells sending out signals to say, hey, something's crazy is going on. And for that to work, you have to have some sort of initial sensor that recognizes something's happened. For instance, let's take the example of you scraped your knee, the damage to the tissue actually will release things that then are a signal for your white blood cells and so on to come to the scene. And as that happens, then more signals are sent by the cells that arrive on the scene, and that signals for more things to arrive. So we get kind of this buildup of cells and fluid and things coming to the area to try to deal with whatever's happening. So that's inflammation. Now the MAC attack, as I told you, is this formation of pores. So we make holes in whatever we're trying to kill. And opsonization, of course, is covering something in antibodies. And so those are basically the three ways that our innate immune system has to protect itself once we've gotten past this barrier of skin and mucous membranes. How do you send out this signal? As I said, there are three ways: classical, alternate, and lectin. So what is the classical way? And again, when we talk about this, what you'll recognize is that although we're talking about the nonspecific innate immune response, there are direct ties to the acquired immune response. So the classical way that you set complement in motion is with an antibody binding to its antigen. We're going to talk about what that means more specifically, okay, in a second. But it is part of the acquired immune response. The acquired immune response is what is making antibodies, and antigen, something that whatever foreign thing is coming into your body is going to have stuff on it that does not look like our normal body cells. There are going to be things poking it out of the virus or out of the bacterial cell wall or out of the protozoan parasite. There are going to be things on the surface of it that look weird to our immune system. And there's going to be an antibody that recognizes that, binds to that part of the thing that looks weird, which is the antigen. And if that happens, in addition to all of the events we'll talk about in acquired immunity, which has to do with making more antibodies and remembering that you saw this thing, also that antibody antigen binding is a signal to start the classical complement pathway in motion. Now the alternate pathway is initiated by something called BPD proteins, which also float around in the blood and also recognize weird things, basically. You can read more about that in your textbook. We're not going to cover it in great detail now for the interest of keeping this overview short, okay? But hopefully that gives you a sense of what things to delve into. Now the lectin pathway is triggered by the recognition of mannose. Mannose is a sugar. Okay, it's a carbohydrate, and it's a sugar that's frequently found on the surface of bacterial cells, and so detecting mannose is a signal that something is in the body that shouldn't be there. So now we're going to go into a little more detail about these things. And as I mentioned, the different components of the complement pathway some of them have specific roles. So we're going to talk about a few of those before we get to what C3 does, because that's really more of an upstream activator. So inflammation, the complement proteins involved are called C3A and C5b. Now what that should immediately tell you, hopefully, since you remember that I told you that complement proteins begin as inactive, large inactive things, is that when you cut the C3 in half or you cut the C5 in half, then it has two pieces, and we call those A and B. So part of C3 is involved in inflammation, and part of C5 is involved in inflammation. Now, when we get to talking about the MAC attack, what's involved in that? That is C5b forming a complex with C6, C7, C8, and C9. So all of these things get together and they form basically this tube and they punch holes in things, okay? Now, how does opsonization involve the complement system? C3b, so the other half of C3, that's involved in opsonization. So that's sort of part of how this whole thing works in terms of just which players are involved. And again, the details of how, what's involved in inflammation, there's a lot to know, so you should read your textbook and become familiar with that. But let's just move on to the activation of these three alternate pathways. So how the complement system is set in motion, which all of those three things happen. However you start the process, then you basically cause, it doesn't matter which general tells you to go into the fight, you're going in, and all the things are going to happen. However, there are some differences in terms of how the pathway is activated. So let's talk about the classical pathway first. And again, this relates to the acquired immune system. So in the classical pathway, we have some kind of bad guy, okay? And along comes an antibody that recognizes something that's sticking out of the surface of the bad guy. So there's our antibody. And usually, pretty much without fail actually, the things that are sticking out of the surface of cells, regardless of whether they're pathogens or normal human cells, they're repeated. There's lots of them, okay? So if there's one of those, there's going to actually be lots of them. And that means that lots of these particular antibodies that recognize this particular thing are going to be coating the surface of this microbe, or whatever it is. Now, this is recognized by a complement protein called C1. Okay, so C1 sees things that are opsonized, covered in antibodies, and it basically recognizes that, and that causes C2 and C4 to be activated. Okay, so C1, if it's activated, it causes C2 and C4 to be activated. What happens then is that C2 splits in half, and C2b is involved in the next step. And C4 also splits in half. Okay, and we see all of these, basically, in order to become active, these cut themselves in half. Now what happens next is that C2a and C4b get together, and together they activate C3. Okay? So let's sort of just revisit how this is working. We have a pathogen, it's got something on the surface that can be detected by antibodies, which are part of the acquired immune system. But that's generically recognized no matter what antibody this is, no matter what pathogen that is, no matter what the signal initially was, that's recognized by C1. That activates C2 and C4 and basically cuts them in half. Now we have 4 pieces, and one piece from each gets together, and together they activate C3. Okay, so they cut C3 in half, so now C3 is in half. And you'll remember, hopefully, from our last list, so let's just go back and have a look, look, there's half of C3 involved in inflammation and half of C3 involved in opsonization, which is actually an antibody binding to its complex. So, you know, there's a feedback loop here also. Now, it's important to know that, as I already mentioned, C3b is involved in opsonization. C3a is involved in inflammation. And also, C3b has a role in something called cytolysis, which again, I'm not going to spend a lot of time going into here, but you can read about it in your textbook. All right, so that's the classical pathway. Now let's look at the alternate pathway. So here is a microbe of some kind. Again, we'll give them a different name just for fun. And the thing about this is going to be kind of abstract. We have to sort of cast our minds back to a number of concepts. One of the things I need you to sort of keep in mind as I go through this is the concept of the different kinds of biomolecules that we talked about in chapter 2. Okay, so you'll remember things that can be carbohydrates, lipids, sugars, and we talked about, you know, where those things are found in the cell and what they do. Sugars can be fuel. They can also decorate the outside of a microbe or a body cell, and they're part of the ID, the identification, the signature of that cell. Lipids, of course, are in the plasma membrane. They also can be fuel, and they also can be decorated with sugars. And the way all cells have these features, this is just true. And the particular sugars and the particular way the sugars are organized and which lipids they're attached to vary a lot. So it actually can be a very unique sort of specific signature on a cell. The kinds of carbohydrate-lipid combinations that you see on bacteria, for instance, are significantly different and noticeably different than as compared to human cells. So even though what I'm about to tell you, this BPD protein pathway doesn't recognize very specific things. It recognizes patterns of things. It recognizes, you know, strings of sugars that are very unusual that you would not find in the human body normally. So on the surface of this bacteria, there is some kind of, you know, lipid in the membrane, and also, you know, in the cell wall, there's lipid, for instance, in the lipid polysaccharide layer of gram-negatives, right? And this lipid has sugar on it, decorating it. That's very common, as I said, for all cells. But this particular arrangement is unusual, and it's recognized by proteins that we call BPD that are just floating around. And really, they just float around in the blood with nothing to do all day except for bind to weird things that are patterns of things that are unusual. They're not very specific. And so how can I sort of make this a little bit easier to understand? When we talk about acquired immunity, the antibody involvement is the antibody is going to bind to something very, very specific, that's so specific that you'd pretty much only find it on, say, one kind of virus, and maybe even only one strain of that virus. It's an incredibly specific fit. Whereas BPD is binding to unusual things that are found sort of generically on the surface of a lot of bacteria, for instance. So their BPD binds this thing, and the binding to this unusual thing that it's been looking for is a signal. But it signals to C3 right away. Okay, so it doesn't even bother going through all these other proteins. It just goes to C3. C3 gets cut in half, okay? And, you know, we just talked about all of the consequences of that. All right, so basically everything is set in motion when C3 gets cut in half. We start the entire thing. Now, the lectin pathway is, again, it's, in a sense, it's similar to the other forms of activation, because let's draw our, invader. We'll call him something else. Call him Z, as they say in my home country of Canada, or Z, as you like to say here in my adopted country of the US. So there is this microbe Z, and as I mentioned, there are mannose on the surface, okay? And there's so much, it's so common to see mannose on the surface of bacteria that it is a more distinct signal than whatever this carbohydrate-lipid combination is that BPD recognizes. Mannose is bound by lectin proteins. Lectin proteins are just proteins that like to bind the sugar mannose. And what happens next is that this activates both C2 and C4. Now, hopefully, this is starting to look a little familiar, okay? The lectin pathway, it has a different trigger than the antibody-antigen classical pathway. And it is a little different because it doesn't go through C1. Instead, it goes straight to C2 and C4. What happens next is very predictable. C2A, C2B. We also get C4 splits in half and becomes active. C4B, C4A. And as we know, C2B and C4B get together. And they together, what do they do? So ask yourselves, look at this picture and ask yourself this. What is going to happen next? What do you think? Look over here. When these guys got together, they activated C3. That is exactly what happens. Okay, so C3 splits in half. We get all of the effects that we have been talking about. Okay. So that was innate immunity in a very quick nutshell. Now, as I've mentioned several times, there are links to adaptive immunity. They are not independent branches of the immune response, but they're sufficiently different that we talk about them separately. We're now going to move on to adaptive immunity. That is the specific branch that responds to a particular pathogen as opposed to responding to whatever foreign things come into your body. They're referred to as the third line as opposed to, remember, the first line, which was what? That was the skin. And also, the mucosal secretions and the normal microbes that inhabit our bodies, and as opposed to the second line, which included antimicrobial proteins like complement and BPD and stuff like that, we have this third line of defense. And it relies very heavily on two cell types, B cells and T cells. And for sure you've heard of T cells because T cells are affected by HIV, so they're very famous. And B cells, many people have heard of those, but take a minute to think about what they do. Okay, the answer is that they are the ones that make the antibodies. So now let's talk about a couple of terms that you kind of need to memorize, and I'm sorry about this. It's just one of those situations where science has a history, and unfortunately, some of the historical terms are still in use, even though they don't necessarily help you very much. They don't actually help you as much as, say, calling something a B cell or a T cell. But I'm going to explain these terms and how they came to be. So there's what we call the humoral part of the adaptive immune response and the cellular part. Okay, and this is confusing because I just told you that the adaptive immune response relies on two cell types, right? So why aren't they both cellular? That's weird. And why have I designated the humoral to be the B cells and the cellular to be the T cells? Well, it's because a long time ago people referred to the body's fluids as humors. And B cells, in fact, are in the body's fluids, and the most important thing they do is make antibodies, and those antibodies are definitely in the body's fluids. So in that sense, they're humoral, okay? They're really closely associated with those bodily fluids. On the other hand, we have cellular, which are just T cells. And that's not to say that T cells aren't found in the blood or the lymph, but they're, I don't know. I just recommend that you memorize that this is what the distinction is. The B cells are humoral and T cells are cellular. Now, the way you distinguish between these two cell types / two types of immunity, humoral and cellular, or B cells and T cells, has to do with where they are born, where they mature, and then how and where they learn the things they need to do in order to be a proper part of your immune system. So, both B cells and T cells are actually born in the same place. So I'm going to give you a second to think about what you know about blood cells and where most likely those are going to be born. That's right, they are in, sorry about that, the bone marrow. So they're both born in the bone marrow. Your textbook goes into basically hematopoiesis, which is the generation of new blood cells. There are ancestor sort of stem cells of blood that can differentiate into all the different kinds of blood cells, and there are many, okay? There are lots and lots. Many of them are involved in innate immunity. Some of them, such as B cells and T cells, are involved primarily in adaptive immunity. Now, the big difference happens once they're born in the bone marrow. One of these cells stays there, and one does not, okay? So, again, think about what you might predict. I'm going to tell you now, and there's a very easy way to remember this. The B cells mature in the bone marrow. And it's very easy to remember that because B cells start with the same letter as bone marrow. On the other hand, T cells are mature in a different organ that you've probably heard of, right? The thymus. And again, this is very easy to remember. T cells for thymus. That's where they mature and where they are educated. And now a couple of things we need to say about what they're learning. First of all, the naive cell, what makes it special is something that's randomly generated. So I'm going to explain what that means in a second. But I want you to sort of remember that B cells, what makes them unique, what makes each B cell its own thing, is that it makes an antibody against something very specific. Now you have many, 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 many different B cells, okay? And their main distinction is just the particular target of their particular antibody. So on any given B cell, okay, they're going to have on their surface an antibody, and they're going to have an antibody that binds very specifically to just one kind of thing, and that is going to be covered, they're going to be covered in that one antibody, okay? Now, next door neighbor, B cell over here, might have a different kind of antibody, and it's going to be covered in those antibodies. So this guy's always going to be looking for triangles, and this guy's always going to be looking for squares. But the reason that they're looking for those things is something that's randomly generated by shuffling DNA around. So in the beginning, you get all of these B cells that are randomly generated. Okay, so they're generated against many, many different possible things, but not in an intentional way. Okay, it's not like we know ahead of time what we're going to see. We just generate lots of things so that you can be prepared for most anything the world can throw at you. Now, at this point, I'd just like to say that the same thing is true of T cells. So antibody binding, the antibodies that a B cell makes, that's what makes it unique and special. And that's in its naive state. It's already got this randomly generated looking for something. What T cells have that's randomly generated that will sort of distinguish them from other T cells is called the T cell receptor. We're going to come back to that. That's also randomly generated. So we have these B cells. They have these differences. That's in their naive state, uneducated, okay? When they're activated, that's when they begin to make a lot of antibodies. And we're going to talk about the process of their activation and what the outcome is of that. But I just want to point out that what that means is that this B cell, okay, is looking for triangles. In its whole life, it might go its entire life and never run into the thing that it's been designed to look for. and it dies. Unfulfilled, so sad. On the other hand, this guy might be luckier. Okay, maybe he runs in his square. Maybe he finds that square very early on. And the finding of the thing that it's looking for, that's the activating event. It's activated by binding to whatever antigen it's been looking for. And we'll talk about what activation is and how it works. Now, on the other hand, T cells that have been randomly generated, what they're learning, this is actually fairly complicated. I'm going to gloss it over and simplify it extremely to the most important thing. Okay, what they learn is what not to respond to. Okay, now this is a euphemistic way of saying that here's this T cell. Okay, I'm going to draw a T cell. And here is its T cell receptor. Maybe the T cell receptor is looking for something that's circular. Now, it may go its whole life without finding that thing. It might find it. But if it's unlucky enough to have been randomly generated to respond to something that's normally found in the human body, so let's suppose that here's a liver cell, okay, and all liver cells have something round sticking out of them, then in the thymus, T cells are tested. So that they go to the thymus and they're tested against all of, this is early in life, they're tested against all the things that are normally found in the human body, including things that might be circular that found on a liver cell. And those T cells, if they respond to something in the human body, they are killed, okay? So they don't really learn what not to respond to so much as if they respond to the wrong thing, they're killed. And they're eliminated from the population, okay? So that's, they start out as randomly generated, some of them are killed, and then the rest of them also may at some point in their lives run into what they're looking for and become activated. And again, they're going to be activated by an antigen. We're not going to really spend a lot of time talking about antigens in a T cell context. It's going to be more broad, as you'll see. Okay, so let's talk now about, so this is sort of how the B cells and T cells arise. We need to now discuss their activation and function before we talk about how that happens. We need to talk in more general terms. So B cells, as I mentioned, they're randomly generated and they're naive. When they haven't met what they're looking for yet, they're considered to be naive. But once they're activated, so once this B cell, so let's draw our little B cell again, the lucky B cell with the square, and say it finds its square, okay? Then it becomes activated. And the process of activation causes two things to happen. It causes the B cell to change, what we call differentiate, and it can change into a plasma cell, and a plasma cell makes antibodies for a living. That's all it does, okay? So this antibody here on the surface, once it's activated, if it's a plasma cell, it's going to make lots and lots of those and secrete them, okay? So they go around floating in the bloodstream. Some of these are going to differentiate into memory cells. So they're going to get basically a long life. Instead of pumping out lots of antibodies, they're given the gift of long life so they can hang around in larger numbers. And if you ever have a pathogen that comes into your body with this particular signature, whatever this antibody was responding to, you're going to have a much quicker response. And that's sort of the basis of our secondary immune system. Similarly, cellular, okay, the cellular part of our, not the humoral, but the cellular, the T cells, we need to talk about two different types. Now, I'm oversimplifying here because there are many different types of T cells. And I'm not going to talk about all of them right now. I'm going to talk about the two most important ones, T helper cells and T cytotoxic cells. Now, T helper cells are very important. They're part of the activation of B cells, and we're going to see that in a second, but let me sort of give you an idea, sort of a preview of why that is important. Basically, B cells are wandering around looking for something, and unlike T cells, they haven't been educated in the same way, right? They haven't been told not to respond to the body, and so if they respond to something, then they can't do anything about it unless a T helper cell comes along, looks at what they're responding to and says, oh yeah, no, that is weird. You're totally right. Let's make a bunch of antibodies against it. And so T helper cells are important for activating B cells. They're like a second opinion, Okay, once T helper cells are activated, they also, some of them become part of the cellular memory. So some of them are given the gift of long life and will remember what they saw, so that you can have that immediate immune response if you should ever encounter, whatever virus it is again. On the other hand, T cytotoxic cells, they have a completely different job. Unlike T helper cells, they're not involved in killing things that come in to the body that shouldn't be there. They are involved in killing us. Okay, which is kind of funny when you think about it, right? Because we just talked about how T cells are educated not to respond to body cells, which is totally true. But our body cells aren't necessarily well behaved for our entire life. I'll give you 2 examples. Okay, so for instance, let's suppose one of your body cells develops cancer. One of the things that happens in a cancer cell is that the kinds of proteins it makes change. And when that happens, it's actually, we'll see how this works in a second, but the protein complement, the sort of sum total of the proteins inside of any cell, is actually displayed on the surface of the cell in something called the MHC1. And that is a place that TC cells go and look in. And if they find something in there that shouldn't be there, then they send up a signal. Now, let's just sort of take a step back and think about how that works and why it works. So T cells, helper cells, and cytotoxic cells are all educated not to respond to the body cells. So if a TC cell goes and looks at all the body cells and sees that everything is normal, it's not going to do anything. It's not trained to respond to those normal things. But if a TC cell is floating around and it sees something on the surface of a body cell that looks weird, it hasn't been trained not to respond to, if that's not too many double negatives for you, then it's going to initiate a cell death program. So here's our TC cell, and when it's activated, okay, so now let's draw our, and again, it has a T cell receptor. I'm going to draw one that looks like that. That's its receptor. It's looking for something that looks like this. Something weird, something that the body doesn't make. And if it finds it and it becomes activated, it differentiates into a cytotoxic T lymphocyte. And so in that new form, it has several superpowers, one of which is to make perforin, and the other is to make granzyme. And these are both enzymes that are secreted, and they go and they punch holes in the body cell that is displaying this weird thing. And again, when you punch holes in things, that is not a good thing for the cell that's got holes in it. So basically the job of TC cells is to look for body cells that have gone bad. Now I was going to give you a second example of, cancer is one example. So if our cells become cancerous, they start to change the proteins they make, and that's detected as weird. But a virus infecting your cells is another example. Okay, so think about this and think about why it's important. A virus gets into your body. Now, it's floating around in the bloodstream, for instance, let's say, and in that milieu, it might be detected by a B cell, and it might actually initiate an immune response. But let's suppose it's lucky or sneaky, and it gets past that defense, and it gets into your body cells, and then it starts to do what? Now think about what viruses do. They have a genome. That genome encodes proteins, viral proteins, that are helpful for the virus to replicate, to make more protein particles for its coat, and to replicate its genome and assemble new particles and get out again and make the rest of your body sick. So essentially, it's tricking your body cells into making its own proteins. And the only defense we have against that is that every day, every body cell takes a sample of the proteins inside of it and sticks it outside of the cell for display, and that is perused and checked by the TC cells. And if they see something weird, such as a viral protein, then that triggers the TC cell to turn into CTL and kill that cell. There's a couple of things we need. Another, again, it's a little bit more terminology. So something we have to know about the B cells is that they are considered APCs. What does that mean? APC stands for antigen presenting cell. We're going to go over this in a second, but I just kind of want to give you a heads up. Now, B cells are not the only APCs, okay? But they do present antigens, and the picture I'm going to show you will help you, okay? But just bear with us for now and learn this terminology. Now, APCs, whether they're B cells or other kinds of, for instance, phagocytes or whatever, they display MHC2. What does that mean? We're going to talk about what that means. And I want to give you these terms now so that you can hear them a couple of times and they won't be so scary and unfamiliar. But APC cells display the MHC2. Okay. On the other hand, T cells, and as I told you already, T cells have one of the things that makes each T cell its own unique thing is which T cell receptor it has. So T helper cells have a T cell receptor, T cytotoxic cells have a T cell receptor. In addition, all T cells have CD proteins. Now, we're going to talk about those only sort of superficially. I'm going to tell you now what CD stands for, cluster of differentiation. That means absolutely nothing to you. Don't worry about it. It's another protein complex, and it sits next to the T cell receptor, and it helps the T cell to sort of interpret and analyze the world. So we'll see how that works. But what I want you to remember for right now is that in addition to its normal T cell receptor, T helper cells have CD4. Okay, it's a particular form of the CD. And this combination looks inside of MHC2. So this is going to make a lot more sense when you see the picture, but I want to plant these terms, these new jargony terms into your mind. Now, on the other hand, cytotoxic, T cytotoxic cells have a T cell receptor that's matched with a CD8, okay? And that looks inside of MHC1, whereas MHC1 is found on body cells. So we're going to go back to this in a couple ways, but I want you to start thinking about these different terms and trying to remember them. I know they've sort of, you know, MHC1, MHC2, how am I going to remember the difference? That seems like kind of hard. CD4, CD8. Well, if you know much about HIV, which you may already if you're heading into a professional health program, you may have heard something or maybe more than something about it, then you'll know that T helper cells are targeted by HIV and they have CD4 on them. Okay, so that's one way to sort of start remembering that. So let's, again, define and these terms. So MHC, what does that stand for? Major histo, and I can't spell today, compatibility complex. Okay, what that means is that there's a bunch of proteins, and that bunch of proteins makes a complex on the surface of some cells, and we'll talk about which cells and what kind. I already told you that APC stands for antigen presenting cell. Okay, so we can talk a little bit more about this. So that is going to be any cell that, you know, sees and responds to triggers in the environment and then presents them to somebody for review. Okay, antigen presenting cell. For instance, not the only one, B cells. Okay. Now I told you already what antigens are. They're things that antibodies bind. B cells are making antibodies. So what's happening, you can start to sort of piece this together. Antibodies binding antigens, they take that inside. We'll see that in a picture in a second. They take that antibody antigen complex inside, and then somehow they display it for somebody to look at. Who is going to be looking at it? I already told you that B cells need to be activated by talking to, getting a second opinion from T helper cells. Okay. Now, so here's our relevant term relating to T cells, T cell receptor. So again, what's that? Just like B cells have unique antibodies, T cells have unique T cell receptors. And this, of course, cluster of differentiation, which is just a term for a complex of proteins that you don't really have to know much about. It's another complex of proteins, and all you really have to remember is that it's going to, like the TCR, it sits on the surface of T cells. So now we can sort of go into what is going on. Okay, so there's all our different terms. And what I'm going to do now is draw for you how activation of B cells and T cells actually occurs. And what you will see is that there's a fair degree of discommunication between these two cell types. So when you talk about one, it's hard to do that without talking about both of them, actually. Okay. So we need to talk about B cell activation first. I like to talk about B cell activation first because I think that people may be a little bit more familiar with B cells and the fact that they make antibodies. T cells kind of sneak into the picture, and sort of it's a little bit easier way to grasp this material. But you could do it in either order. It doesn't really matter because as you'll see, they talk to each other. Okay, so let's start by drawing over here a B cell. And we're going to make this a naive B cell. Okay, hopefully you can see that in my terrible handwriting. That's naive B cell. So a B cell that doesn't yet, it was born in the bone marrow. Where did it stay? It stayed in the bone marrow. And then, so it's naive, right? So, but it already has randomly generated a specific, just one kind of antibody all over the surface. Okay, so it's got all these antibodies. These are the particular antibody that's made by this particular B cell. Now there's something else that we need to put on the surface, and I haven't really mentioned this before, but it is very important. And so I'm going to make a little room for it. Okay, just going to get rid of that guy. I'm going to move him over there. And here, and this is definitely not to scale, I'm just drawing it big so that we can all see it. That is called MHC2. And I'm drawing it kind of in this semicircle. I want you to think of it like a basket, okay, that it's wearing on its head. And whenever this B cell's antibodies encounter what it's been looking for, that antibody-antigen complex is brought inside, chopped up, and placed into the basket. So let's kind of see if we can draw a picture of that. So what has this guy been looking for? Something triangular. So let's draw a pathogen with something triangular on it. Okay, and this detection event causes this whole thing to be taken inside the B cell, and it goes chopped up into pieces. So even that triangle is probably chopped up into 2 pieces, and those pieces are put into the basket. Okay, so why are we doing this? Here's our foreign object, whatever it is. Foreign microbe, foreign virus, foreign bacteria. Who is looking in the basket? Oh, well, let's draw that guy. Who do you think? Who does this guy need to get a second opinion from? If you said T cell, you're right. Now we know there's 2 kinds of T cells that I already talked about, so if you said T helper cell, you're even more right. And what did we say was on the surface of the T cells? We have T cell receptors. Okay, there's the TCR for this particular T helper cell. And next door is the CD4. Okay. So I'm going to simplify this a little bit, and this is really all you would need to know for this course. If you find this stuff fascinating and you pursue it further, you'll find that there's yet more nuances to be learned. But for the sake of this discussion, what basically happens is that the CD4 recognizes the MHC2. Okay, so that's what allows the T helper cell to dock in the right place on the right type of cell. And the T cell receptor is looking for something triangular in the MHC2, and I've not drawn these to scale, but basically it's been looking for this little piece here. So if it finds what it's looking for in there, it says, oh my gosh, something is happening. It becomes activated and it sends an activating signal to the B cell. Okay, now what happens then? What do we know about what an activated B cell does? Well, that initiates differentiation into either a plasma cell or a memory cell. And T helper cells also differentiate, and some of them become memory cells as well. Okay, so what did the plasma cells do? You remember what they do? They make lots of antibodies. So how is this different from what they've been already doing? You might ask yourself. It's a good question. So here's the naive B cell, and it has antibodies on the surface, okay? But what plasma cells do, and this sort of brings us back to what you may not have actually learned a great deal about, but we will just mention it briefly. The different internal contents of a eukaryotic cell, all those organelles that you get to hear about in excruciating detail in some bio courses, are very important for the plasma cell. So what this cell wants to do in its differentiated form is make a lot of these antibodies, like tons and tons, not just enough to coat the surface, but it's going to secrete them out, and they're going to be soluble and it'll float around in the bloodstream and the lymph system. So it needs to make lots and lots of proteins. Now, what you may or may not know from former biology courses is that the organelles in the eukaryotic cell that are important for making proteins that are going to be exported are the endoplasmic reticulum, the Golgi body, all those little vesicles, but mostly the ER, the endoplasmic reticulum. It's decorated with all those ribosomes, so rough ER, if that is sounding familiar to you. This differentiates into a cell that has lots of rough ER, okay? And it makes lots of antibodies using that rough ER as a factory. And it's going to make this specific kind of antibody here, okay? If it was a different B cell, it would make it a different antibody. Some of them, of course, become memory cells and are saved for future encounters. Now, there's an exception to this, okay? And I'm not going to say how it works really. You can read about it, but I'm just going to make sure that you know that there's something called T-independent antigens. Now, what do you think that means? That means that sometimes a B cell can meet an antigen that causes it to become activated even though it doesn't get a second opinion. And I'm just going to sort of say one example. For instance, there are certain things that have a long body and a lot of repeating elements of the same thing that would trigger this B cell so much that it's just going to activate anyway. So just know that that's the case and read more about it in your textbook. Okay, so there's our picture of activation of the B cell. Now, let us talk about activation of the T cell instead. And I'm not going to talk about the activation of the T helper cell. I basically just did that. You see how I snuck that in there? I snuck in the activation of the T helper cell because it's happening at the same time as the B cell. What we want to learn about now is how do you activate a T cytotoxic cell and turn it into a cytotoxic T lymphocyte. So how does that work? So again, let's draw our naive cell. Okay, so here's our TC. What do we know about it? We know it's got a T cell receptor that's unique to that cell. It's got a CD, but remember, which CD does it have? Do you remember? What CD did the T helper cell have? That was the CD4. So this is CD8. Okay, so what is it talking to? B cells and T helper cells are talking to each other, and in response to some microbe that the B cell is talking to. Who is this guy talking to? What is the purpose of this guy? Okay, yes, if you thought body cell, that is entirely accurate. So let's just draw a body cell. Okay. One of many different ones in the body. Now, what's on the surface of the body cell? I haven't explicitly told you this, but we want to sort of think about what we already know about all these jargony terms we've been learning. The TC cell is going to bind to MHC1 and look inside of that basket. So we need that basket on the body cell. Here it is, okay? But it is MHC1. I've drawn it as a basket, okay? It does actually resemble MHC2, but it's sufficiently different, and the difference has to do with CD8 is going to recognize MHC1, and that's going to guide T cytotoxic cells to the body cells so they can peer inside the basket. And what are they going to be using to look inside that basket? They're going to be using this T cell receptor to look inside there. Now, what are they going to be looking at? Okay, so remember I already told you that all the cells in the body, every day they go and take a sample of the proteins that they have inside of them, chop them up, and put them in the basket. So let's do that. Okay, so let's have this protein here, and maybe that protein that looks like a mushroom. We're going to chop it up into pieces, and those pieces are going to look a little bit different than they actually look like in the intact organism. Okay, they're all those little pieces. It's going to be looking in there for something weird, okay, and it's going to only be looking for things that it has been not trained or not killed because it responded to. So randomly generated thing, that means that your body cells are visited by a lot of different T cells, but it's looking for something square. And in this particular case, you know, I haven't drawn this very clearly, but here's our sort of diamond-shaped guy that's generating these triangles. So they really shouldn't be squares. Okay, let's get rid of that. But, so, you know, normally it's not going to find what it's looking for. That's a normal case. But what if this was a cancer cell or a cell infected by a virus? Let's see what happens then. Well, then the virus might be making, say, this square protein, or the cancer might cause this cell to make different proteins that make something that is, in fact, the right shape for this T cell receptor to respond to. So CD8 grabs the MHC1, TCR peers inside. If it sees what it's looking for, it activates. Okay, what does activation cause? Causes this cell to differentiate into, instead of this nice, happy-looking T cytotoxic cell that's just, you know, like a cop who hasn't, you know, sitting on the side of the road with his radar gun falling asleep over a sandwich because, you know, nobody's speeding. Suddenly, oh, sirens on, tracing someone down on the freeway. If this has recently happened to you, I'm sorry for the trauma that I'm causing you. All right, I have not been pursued at high speed by police, just in case you're wondering. So this CTL is now able to release. Remember, it's going to make perforin and granzyme, and these are very damaging. They're going to go punch holes in this cell. I'm drawing holes, but you know, really what's happening is they're punching holes, so just boop, boop. Okay, so this cell is going to be really destroyed. Now, that's CTL activation. One more thing I want to say. Here's another what if. What if the cancer or the virus caused this body cell, okay, to be so weird that it stopped making an MHC1. So there's no MHC1 to look inside of. I mean, that's clever, right? If you were a virus, this would be perfect, because then you can make all the weird proteins you want and no one will know. So in that case, there is a cell type called natural killer cells, okay? And they have very similar capabilities to CTL, but they only attack and specifically look for cells that don't have an MHC. So they're going to chase after a lot of things, right? A lot of things that shouldn't be in your body won't have an MHC, obviously, but if your body cells forget that they're supposed to present themselves for inspection, those guys will make sure that nothing gets past, okay? So again, they will basically, punch holes in the cell and it dies. Okay, so there is our T cell activation. Now, there's one more video that I'd like you to watch. And I think it's a nice illustration of the adaptive immune response. So let's have a look at that. So here we are. Where are we right now, would you say? If you said that we are inside a blood vessel, you're right. Now, as we watch the blood cells streaming past, we try to identify them. What are these red things? Red blood cells. What are these white things? White blood cells. I know that's really hard. And then suddenly this guy appears. Okay, who the heck is that? This is, something foreign in the body. Maybe it's a bacterium, a virus. And there are these things poking out of it, okay? And this is what I'm talking about when I say antigen. There's these things poking out of the microbe that are visible to our immune system. So there it is, and what's happening now? It's docking on something, it looks like. Multiple things are docking. What is happening? What kind of white blood cell? Look at the difference in scale. This is where all of your background information should come in handy, right? You've learned what size different cells are, and you know roughly that eukaryotic cells are between 10 and 100 times bigger than prokaryotic cells. Bacteria, prokaryotes, and viruses are even smaller than that. So here's this giant white landscape, looks like the moon, and that is in fact a white blood cell. And all of these things that are docking on it, these would not be normally found in our body. Now, what are they docking on? Ask yourselves that. Oh, and looks like those exact same antigens are being coated with antibodies. It's the same antibodies that are found on the surface of this white blood cell, but just in their secreted form, okay? So now we have, what's this? You're coated in antibodies, you're a target. What is that? That's opsonization. This is, a phagocyte that's just recognizing opsonization. It doesn't care what antibodies are there, it doesn't care what this particular microbe or whatever is. It's just like coated in antibodies, must get rid of it. Okay, so again, now we're sort of reaching into, now this is back to the innate immune response, the non-specific immune response. Grabs it, takes it in, eats it. Gone, okay? So I think that's kind of a nice illustration of both the power of the acquired immune system and how it is integrally linked to and works with the innate immune response. All right. So that's all I have for you today on the subject of the immune response. There are a couple of extra things that we need to talk about before we close this video. And that's just to give you a little bit more perspective and knowledge of some of the terms that we've been using, kind of throwing around as if you knew what they meant. And so I'd like to talk about those now. And they are antibodies and antigens. Okay, so let's talk about antigens first. Okay, so if this is some kind of microbe, let's say, and like any cell, it's got things poking out of it. Okay, then this thing that's recognizable by the immune system, that is an antigen. Now I'm going to introduce one other term here. epitope. What does that mean, and what's the difference between antigen and epitope? If you take immunology any further, you'll find that it's really important to know this. Basically, here's the antigen, which is the whole thing that can be recognized, and the epitope is the particular piece of it that the antibody binds to. Okay, so let's just, for example, let's suppose there is a B cell that makes this kind of antibody, and that can bind here, okay? And a different B cell, which we'll diagram in a slightly different color, can make a different antibody, okay? And so that can bind here. They're both binding to the same antigen, just different epitopes on the antigen. Now, this is a really good time to segue into a little bit of a discussion into the different kinds of antibodies, because hopefully, you know, already sort of intuitively you understand that they're not all the same. Let's think about what we know already that suggests that. When you looked at that video, you saw that the white blood cell had antibodies on its surface that were sort of poking out. And then it also was apparently making, or somebody had made, the very same antibody, binding the very same thing, but in a secreted form. So, what kind of antibodies are there? There are 5 different kinds of antibodies. They're called IgGs, IgMs, IgA, IgD, and IgE. So, what distinguishes them has to do with their structure, and we'll talk about what that means in a second. They're sort of their arrangement with respect to each other, and where they're found and what they're for. So, what is their, maybe another way to say that would be, what is their organization? IgGs are always monomers. What does that mean? That means that they look like this. They have a stalk, they have 2 arms, and the arms at the ends is where the antibody is been shuffled to have some unique specificity, and where it binds its epitope, okay? And when I say monomer, each antibody has 2 arms, so it has 2 opportunities to bind something. And so it has, each monomer has sort of 2 spots to bind. That's a monomer. That's sort of the basic smallest unit of an antibody that you can get. Where do you find these? These are going to be those ones that are secreted. So once you make that plasma cell and they're making a whole bunch of secreted antibodies, those are the ones they're making. And they're found in the bloodstream. And in that video that you saw, those were the ones that were sort of floating in out of nowhere and coating that microbe. Now, what are IgMs? They're found in pentamers. What does that mean? That means there's 5 of these guys. So I'm going to draw it not to scale, a little bit smaller, just so we can fit it all in. So they're all kind of tied together at their ends. And that means that there are 10 spots to bind. Okay. Now, why would you want that? This is going to be a super efficient way to catch bad guys, to detect them. We find these on the B cell surface. Okay. So we're giving that B cell the best possible chance of finding what it's looking for. IgAs are dimers. So basically, here's the, whoops, violating my own conventions. How will you ever learn? There we go. 2 antibodies back to back. They're joined by a special bridge that talks about that in the textbook. It's not that important. But basically, there's a dimer. So there's 4 spots to bind. These are found in secretions, okay? And so you can imagine that they're in the fluid, but it's a sticky fluid, so we have a few more spots to bind. IgEs are always monomers, right? So they look like this. And they're found on mast cells and cells involved in the allergic response. And then we finally have another kind of monomer called the IgD. And those guys are found again on the B cell surface. Now, how can you possibly remember all these things? I'm just going to give you my silly mnemonics, and hopefully that will help you, and I'm sorry if it doesn't. Otherwise, you're just on your own. You'll have to memorize it, because you do need to know. I remember IgGs as being general, because when I think of antibodies, I always think of those ones that are floating around in the bloodstream. Okay, so those are those secreted ones. They're floating around. IgM, these are the biggest ones. These are those pentamers with 10 spots, and I think of those as mega, huge mega. IgA, this dimer in secretions. I think of that using the word achu. Now, this is going to be a little bit confusing because of IgEs, but hopefully you can keep them separate because, you know, it's in your nasal secretions, for instance. IgEs, I remember because they're involved in allergies. So I'm going to spell that in a weird way to make that a little more clear. And IgDs. I don't really have a special technique for remembering that, except that's the one that's left over. Okay. So those are your different kinds of antibodies. All right, that's all that we need to talk about for this small mini lecture on the immune system. Thanks for joining me, and good luck with all your studies.